So thank you for that very kind introduction. My lab focuses on learning about interspecies interactions in the human nasal microbiota. And we do so with the long-term goal of identifying bacterial strains and bacterial-produced compounds from human nasal microbiota that will lead to new therapies to prevent and treat infections in people. And we've been particularly focused for quite a while now on learning more about how to prevent infections by two common human nasal pathobionts, Staphylococcus aureus and Streptococcus pneumoniae. Overwhelmingly, the vast majority of the interactions between these two organisms and us as their host is that they're harmless members of our nasal microbiota. However, they both have the genetic capacity to cause severe, even deadly infections, both in people who are otherwise generally healthy, as well and especially in people who are medically vulnerable. So during my talk, I will often call Staphylococcus aureus, and Streptococcus pneumoniae, pneumococcus. Because these are so clinically important, there's a large epidemiologic literature produced by researchers from around the world, other than us. And I want to just review some of that literature and some key points with you all. So globally, worldwide, Staph aureus and Streptococcus pneumoniae are the second and the fourth leading bacterial causes of death. Their drug-resistant forms are the second and fourth leading causes of death due to infections caused by antimicrobial resistant organisms. So these are very clinically important organisms in terms of the morbidity and mortality for humans. Because of this, there is a large epidemiologic literature about risk for infection, ways to mitigate that risk. And the next couple of slides summarize data from a number of labs. So the first thing that's in the literature is that nasal colonization by either aureus or pneumococcus is a risk factor for infection by the colonizing bacterium at a distant body site. For example, there are now multiple studies over a period of more than 30 years showing that Staph aureus infection isolates, so the Staph aureus from an infection, from an invasive infection, will match the patient's nasal Staph aureus strain about 80% of the time. So when we get infected with Staph aureus, it's almost always the Staph aureus from our nasal microbiota. There are also multiple studies that show that if you can prevent or reduce nasal colonization by either one of these pathobionts, you decrease infections at those distant body sites, so you decrease invasive infections. So as a clinician, I was familiar with this epidemiologic research and also the more recent epidemiologic research. And one of the questions I began to wonder is, why are only a subset of people colonized with these two species? So on any given day, about 30% of the world's population has Staph aureus living happily in their noses. And over 40% of kids under 7 worldwide have nasal colonization by Streptococcus pneumoniae. Now, to colonize us, they have to interact with us as a host. So there's a beautiful twin study from Paul Anderson about 12 or 13 years ago, where they showed that Staph aureus nasal colonization was primarily an environmentally determined trait, which means that variation in host genetics plays only a small role in whether someone has aureus in their nasal microbiota or is free of it. So then, in addition to interacting with us as their host, which is a very important area of research, they have to colonize and then invade and set up residence, right, in this pre-existing microbial community. And, often complex multi-species microbial communities will resist invasion if possible. So collectively, this background information in the literature both supports and underlies the central hypothesis that drives research in my lab, which is that there are beneficial nasal bacteria that are capable of inhibiting aureus or pneumococcus growth or virulence and thereby protecting us. At this point in microbiome research, we actually know what the common nasal species are. and we have some candidate organisms. And one of the key things that we've learned in our own microbiome research is that we should be able to model human nasal microbiota in the laboratory with consortia of a small number of cultivated species. So human nasal microbiota has a very uneven community structure. And what this means is that for over 90% of adults, 90% of the bacteria you can detect belong to 2 to 10 species. So there are many bacteria there, but numerically dominant species are a small number. These species tend to be drawn from about 20 very common species, 19 of which have already been cultivated. And for mechanistic studies, a number of these are genetically tractable already. So from my mind, this makes this a very nice human-associated microbial community for doing mechanistic interspecies interaction work. And so my lab has been identifying factors that shape the human nasal microbiota through microbe-microbe interactions. This schematic illustrates all of the interactions that we have published on to date. A red bar is a behavior modification. No, sorry. Whoa. A red bar is an inhibition. The orange arrow indicates a behavior modification where one organism changes its behavior in response to another. And that blue arrow indicates a growth enhancement, where we've teamed up with chemists and actually identified the compound responsible for the interaction. I've added the structure to the schematic. So this is cool, and it is the tip of the iceberg because there are other labs that have worked on identified other compounds, and because there's just so much more to learn about this microbial system. However, almost all of this work in my lab has been done on agar medium. And what I really want to understand is what happens in the human nasal passages. Are these interactions occurring there? And what other interactions are occurring? And I'll remind you that human nasal passages are coated by 2 distinct types of epithelial surface. So the nostrils or the nares is actually the opening. And when you go into the nostrils, you enter what's called in English the nasal vestibule. And that's lined with a skin type epithelium that you can see if you look in the mirror. A little further back, it transitions to a mucosal surface, and this is the nasal respiratory epithelium. It coats the majority of the nasal passages all the way back to the nasopharynx. It is mucus producing, and it's where you stick the swab if you're going to do viral testing. So one of the things I'm super excited about in my lab in the last couple of years is this wonderful collaboration with the Baylor College of Medicine 3D organoid core run by Sarah Blute and Dr. Tony Piedra's lab. And in this collaboration, my lab's role has been to use and develop protocols for human nasal epithelial organoids that the core differentiates at an air-liquid interface and that we then use to model bacterial nasal microbiota. So to just give you a little background on these human nasal epithelial organoids. So the core will harvest cells from donors, from donors using a mucosal swab and a wash. And among all the cells they get, there's a small population of tissue-resident stem cells. Because these cells have stem-like properties, they survive as the other epithelial cells die. They can be propagated, they can be passaged. They propagate as these little 3D balls of cells, and they can be frozen. So each donation becomes a long-term experimental resource where the same strain can be used over and over again in experiments over years. When we use them experimentally, they're differentiated from these 3D organoids into a 2-dimensional, onto an air-liquid interface where you have air on top, basal medium on the bottom, and then the expected cell types that mimic human nasal respiratory epithelium. And there's some beautiful work from Tony Piedra's lab really showing that the expected cell types are there in the organoids. So I'll just walk you through this cross section. So this is a cross section through an organoid grown in a transwell. And starting at the top, you have air. This is a PAS stain, so the mucus is going to be in magenta or pink. You have a robust mucus layer on top. This fringe of magenta across the top, those are the ciliated apical cells. And in the live organoids, the cilia beat. You can actually see the cilia beating circulate the mucus within this little transwell. The goblet cells are shown here. They're full of mucins, and so they stain pink. And then you have your basal cells. So I want to introduce you to one other bacterium before I talk about our colonization experiments. And this is Dolosa granulum pigrum. It is a common member of the human nasal microbiome. The first time I cultivated out of a volunteer's nasal passages, this is what was known about it, that it's very rarely associated with disease. That is still true. It's a lactic acid producing bacterium. It's a gram-positive. It belongs to the family Carnobacteriaceae. And that Dolosa granulum pigrum strains have broad antibiotic susceptibility. So some things that we've learned about it since then. So one, we've learned that Dolosa granulum pigrum is negatively associated with Staph aureus in adult nasal microbiota. So these are data from our reanalysis of the V1, V3 sequences from 210 adult nostril samples in the HMP1. And what you see here is that if there's no aureus, you have higher relative abundances of D. pigrum. This corroborated data that had been published by Cindy Liu and Paul Anderson in a cohort of older adults in Europe, where in their study, higher levels of Dolosa granulum pigrum was the single best predictor of being free of aureus nasal colonization. There have subsequently been other groups and other studies that have shown this inverse or negative association. When I see this kind of association in vivo data in a microbiome study, what I think is, well, what's the simplest hypothesis? So the simplest hypothesis that would account for this relationship is that one of these bacteria antagonizes the other, or at least one of them antagonizes the other. To date, we haven't found aureus antagonizing Dolosa granulum pigrum in our assays. However, we also haven't looked as hard for that. The first 10 strains of Dolosa granulum pigrum we tested, and those are shown here as spots on the left in each of these images. We spot the Dolosa granulum pigrum. It grows slowly, so we have to wait a couple of days, and then we spot aureus next to it, and each of these strains produces some amount of an anti-aureus activity. With our own Dolosa granulum pigrum nasal isolates, so things only that we know are nasal isolates from healthy people, over 60% of them have an anti-aureus activity in this kind of assay. So because of it, we're very interested in aureus's behavior in the nasal microbiota. So some of the things that we have learned about D. pigrum. So one, from studies from multiple other groups across multiple other continents, we've learned that D. pigrum is associated with a healthy cohort in multiple nasal microbiome studies. From our own strain genomes, we've learned that there's no predicted virulence factors. Similar strains have circulated over a 20-year period in the US. Dolosa granulum pigrum has a highly conserved chromosomal organization, and it has robust defense system against mobile genetic elements. So characteristics that you might expect in a potential mutualist. Okay, so shifting back to the nasal organoids, these data show monocolonization by 3 different species: Staphylococcus aureus, Streptococcus pneumoniae, our 2 pathobionts, and Dolosa granulum pigrum. And what we do is that we colonized with a fixed number of bacterial colony forming units, so we know how much we start with. We do this in a very small volume of liquid to preserve that air-mucus interface, and then we can allow the colonization to go on for different amounts of time. When we want to know how many bacteria are left, we simply harvest the whole thing and quantify CFUs. I'll point out that we do all of our experiments at 34 degrees Celsius. This is the maximal end of the luminal temperature in the human nasal passages. This is work from Elad and Keck a number of years ago. And equally importantly, in my mind, or maybe more importantly, their work from a number of different labs for aureus, for pneumococcus, and also for non-typable Haemophilus influenza, which I'm not talking about today, but 3 very common human nasal pathobionts. These bacteria behave differently at 34 degrees Celsius and lower than they behave at human body internal temperature. So when they're causing an invasive infection, they encounter 37 degrees or higher. There they behave one way. When they're on our surface, they encounter temperatures of 34 or lower, and they behave differently. And so this is a big part of why we do our experiments at 34 degrees C, because we want to model colonization. So these data from my lab show human nasal bacteria monocolonizing the human nasal organoids. The y-axis are colony-forming units of bacteria per organoid. Each dot represents an organoid. We've done this in 3 different cell organoid lines and more than 3 times independently for each. So then they're divided up by bacteria. So the bacteria are color-coded here, and I just want to walk you through that. Our time 0 is the inoculum. So this is what we put on. Then we looked at the number of bacteria 24 and 48 hours later. What we see is there are different colonization dynamics across the different bacteria. What was very important for us is that for each of these species, there is a time point where we can get back at least 10 to the 5th bacterial CFUs. This is enough bacterial biomass for a lot of downstream assays, including doing dual host bacterial transcriptomic studies. So then where are the bacteria? Well, they're in the mucus layer. And I'm going to illustrate this first using dpigrum. So let me just walk you through this. On the bottom, so DNA is stained here with an intercalating dye, and it's colored blue. So you see your epithelial cell nuclei at the bottom. The mucus is stained with an anti-MUC5A antibody. This is the dominant nasal mucin in humans. You see mucus in green. And here you see a bunch of blue in the sea of green mucus. So when you look really closely, these look like little blue dots, like dulosogranulin should look. And we wanted to be sure this was bacteria and not debris from sloughed off epithelial cells. So to do that, we used a method developed by Scott Chmielewski, which was to pre-stain the bacteria with mitotracker red. So the bacteria, live bacteria, they're stained, they stay alive, and then they colonize. And then this is a 6-hour time point. And the mitotracker red survives the fixation so we can see it. But what you see is that there are bacteria here in the mucus layer. We have similar results for Staph aureus and also for Streptococcus pneumoniae. I'm just going to share that with you real quick. So again, there are little dots of blue up here for the DNA for the bacteria. And what you see here are the pre-stained pneumococcus. You see this really pretty lancet-shaped diplococci that's typical of Streptococcus pneumoniae morphology. So the bacteria are living in the mucus layer. So then how are the epithelial cells responding to this bacterial colonization? The first thing we asked about was cytotoxicity by doing a lactate dehydrogenase assay. Lactate dehydrogenase is an intracellular cytoplasmic protein in the epithelial cells that's released if cells are damaged. So the y-axis here is the full change in the LDH from 1 hour before colonization until this is a 48-hour time point. And the gray here is our uncolonized control. So we're looking at how much LDH does the organoid produce and release into the basal medium when it's just by itself. And what you see is a very low level, and it's comparable to what we see with both dulosogranulin and pneumococcal colonization. We see a little higher level of LDH release with Staph aureus. Some are low, but occasionally the aureus is clearly irritating the epithelial cells. And for us, this made sense. This is actually a USA 300 MRSA strain, which tend to be relatively aggressive. Another way we examined how the epithelium was responding to the bacteria was to look at cytokine production. So epithelial cells are really a point of first contact with our microbiota, and they release cytokines that help orchestrate our innate immune response. So for this experiment, again, we're monocolonizing with each of the bacteria, or each different bacterium, and we're using dead, and here we use gamma irradiated, so that we were trying to preserve the antigenic surface, so we've got dead or live cells. So 48-hour colonization, and then a wash to look at the apical cytokines. This tells us what cytokines are released into the mucus layer, which mimics what the epithelium is releasing into the nasal lumen. Or the basal medium, this is mimicking what the epithelium is releasing into local tissue and into the bloodstream. And what we learned are that the human nasal organoids display general and species-specific cytokine responses. So this heat map, for each of the different conditions, we're looking at the log 2-fold change compared to the uncolonized organoid and the cytokines that it produces. So your eyes, I hope, will be drawn to those two columns of red. That's increased production. And we see that regardless of the bacterium and whether the bacteria are alive or dead, the epithelium, nasal epithelium, is producing an increased level of CCL20. This is involved in interactions with macrophages. There's also increase in the production of GCSF. This is involved in interactions with neutrophils. In contrast to this general response, then we have species-specific response. So if you look at the arrow that I think should be on your left over here, this points to IL-18. And you see only live Staph aureus is inducing an increase in IL-18 levels. IL-18 is involved in inflammasome signaling. So the first step when cells are irritated or damaged is they release pre-made IL-1 alpha. So that's a general, they release it. That it's called an alarmin. That then binds to its receptor, and downstream of that is the production and release of IL-18. And then this is again consistent with this strain and the fact that this strain can irritate the epithelium. What we're seeing over here on the blue arrow on the right side is actually a decrease in production of a cytokine in response to the bacteria. So what we learned is that Delosa granulum pigrum, live or dead, decreased apical production of CXCL10, which is involved in antimicrobial responses. It's been implicated both in antiviral and in antibacterial responses. So now we're looking at the picogram per mil of the cytokine detected in an Illuminex ELISA assay. And the orange arrows show you the level when you have live or dead D. pigrum, and this is the baseline level in the uncolonized organoid. So we also saw an effect of live Staph aureus, but not dead Staph aureus, making us hypothesize that the mechanism may be different. Okay, so we've got this model up and running now. I've showed you monocolonization data. We're now colonizing with 2, 3, and 4 species at the same time. And I want to just give you a sense of the kind of assays we're now able to do using this model. So we've been doing dual bacterial epithelial transcriptional response assays where we can look both at microbes interacting with the epithelium, and we can look with 1 or 2 microbes so we can see if there's an effect of adding a second microbe on either the epithelium or the microbe response. This is some preliminary data from one of our organisms. Our organoid core has now developed a number of infant-derived organoid lines. We've shifted to using these to study pneumococcal colonization because, of course, pneumococcus is such an important pathogen in children under 4 years of age. This is aspirational. Our goal is to begin to use this model to identify colonization-resistant communities, so communities that in this specific human microenvironment of the nasal mucus will resist colonization by aureus or pneumococcus. I think this is going to be a wonderful system for testing lytic phage for potential use for Staph aureus decolonization. And here I'm going to share with you some very preliminary data modeling the killing of this USA 300 methicillin-resistant Staph aureus strain called JE2 on organoids. And this is the log two-fold change in the number of bacterial CFUs when we have just the bacteria alone versus we've added a variant of phage K, which is a lytic or a staphylococcal lytic phage. And here we just have the Staph aureus, but we also have it now co-colonizing with different organisms. And over here, it's one of 4 organisms colonizing. And we see that even with other bacteria present, we're getting specific killing of our Staph aureus. Then another thing that we have been doing is to use TNSeq. So we've used a TNSeq library that was generated by Anthony and published by Anthony Richardson's lab. It's in, again, this same USA 300 MRSA background. It's in a strain called LAC. And we've used this to begin to identify aureus nasal colonization fitness factors. So there are some known fitness factors from studies in animals or studies with cancer-derived cell lines. We wanted to see what we would see in this, what we think is an improved biomimetic for the nasal mucosal environment. And one of the things that jumped out at us from our hits, the hits are shown on the left, is that genes involved in synthesis or use of D-alanine are candidate nasal colonization fitness factors. So that makes sense because D-alanine is very important for bacteria. They produce it, humans don't. So D-alanine is used by bacteria for peptidoglycan cross-linking, which is important for the structural integrity of the cell wall. Many gram-positives, including Staph aureus, will also dealinate their surface-exposed teichoic acids. This is a way to control the surface charge and protect them from cationic antimicrobial peptides. When we look at our data, we had 3 hits in this pathway related to D-alanine. The 2 producing enzymes, as well as this enzyme involved in dealination. But what surprised us when we looked at our hits is that D-alanine aminotransferase, this DAT gene, was our biggest hit. So I'll go to the next slide. When Staph aureus is grown in vitro in rich medium, D-alanine aminotransferase has been shown to primarily generate D-glutamate. So this is a reversible reaction. The D-alanine aminotransferase can either produce D-glutamate or produce D-alanine. There's a really nice paper from Vinay Thomas's lab showing that in culture, it goes predominantly this direction, leaving the alanine racemase, or the ALR gene, which interconverts L-alanine and D-alanine as the major producer of D-alanine. So if you look at the in vitro data, you would hypothesize that the ALR gene would be more important than the DAT gene. So we asked, well, what happens if we disrupt each of these genes with a transposon and then look at the colonization on the organoid? And what we learned is that disrupting the alanine aminotransferase led to an over 3,000-fold colonization defect on human nasal epithelium. So again, our readout are colony-forming units per organoid. This is our wild type. Here is the D-alanine aminotransferase mutant. And then this is the alanine racemase mutant. And these levels are comparable, at least statistically. So in contrast to what we see on the organoid, in complex medium, each of these individually is dispensable and they all have the same growth rate. So again, D-alanine aminotransferase, the DAT gene, is either producing D-glutamate or D-alanine. And because all of our data pointed towards D-alanine, we hypothesized that maybe we could complement this defect by adding exogenous D or L-alanine. And that's shown here, where we do get rescue. So I'll just walk you through this graph. Then this is colony-forming units of the bacteria. This is our wild type. It's similar to what you just saw. This is that D-alanine mutant, D-alanine aminotransferase, the DAT mutant. It has the colonization defect. And now here's our dose response to adding exogenous. So we're just putting a little drop of D-alanine onto the mucosal surface. And what we see is as we increase the level that we add back, we see rescue. So at 15 millimolar of D-alanine, we hit wild type levels of colonization. We also see that we can complement with L-alanine. So L would go through ALR to D. That is gone. And so we're adding D-alanine and we're complementing. Again, this is chemical complementation. We're in the process of building the strains to do the genetic complementation. So our data points to loss of D-alanine aminotransferase as reducing aureus fitness on organoids in a D-alanine dependent manner, leading to the hypothesis which we are currently working on, which is that on human nasal respiratory epithelium, it's D-alanine aminotransferase that is the major D-alanine producer. And we want to confirm that this is so, and then begin to understand why. Why is this so, and why is it very different than what we've learned, what multiple labs have learned in vitro? All right, so I just want to wrap up by reminding you that I think the data I've shared, I hope, has shown you that we use human nasal bacterial microbiota as a clinically relevant model system to identify causal factors that shape microbiota composition and epithelial response. And we do this with a long-term goal of identifying bacterial strains and compounds, as well as more recently, phage, that will lead to new ways to prevent and treat infections in humans. And I want to end by thanking the people in my lab. I have been privileged to work with a number of fantastic colleagues in my lab. This is my current lab here, and past members of my lab, and also our wonderful collaborators and our funding sources. And I'm happy to take any questions.